Hello and welcome to another video. In today's lecture, we are going to continue from exactly the point we left. So please make sure you have watched the previous lecture before you watch this video. In previous lecture, we studied that a zygote was formed as a result of a fertilization reaction. The fertilization reaction took place between a male sperm and a female oocyte, where they both combined to form the zygote. The zygote structure which was formed was a unicellular, singular structure which was called a zygote. Zygote basically means a thing which is made by one cell. Now, in today's lecture, we are going to study that the zygote will undergo a mitotic divisional process called cleavage. And once the cleavage starts, now the zygote will be called an embryo. An embryo means a structure which is formed by many cells. By the process of cleavage, this changed into a two-cell stage, which was about 30 hours after fertilization. This two-cell stage will undergo further mitotic division, which will change this structure into a four-cell stage. Now another mitotic division, and this four-cell stage will be changed into an eight-cell stage. So you can understand that mitosis is happening and the cells are going to multiply on every occasion. Okay, so when the 8-cell structure is divided further, it changes into 16-cell stage, and now when it arises to the 16-cell stage, now this cellular structure is known as a morula. This 16-cellular stage will undergo another process called compaction while it is in the same morula stage. As a result of compaction, what happens is that there are these two cellular masses formed. The one on the inside is known as an intercellular mass or embryoblast, while on the other side or the outer wall or the outer cellular mass will be known as a trophoblast. While all these structural changes are happening inside the morula, now it has arrived in the uterine cavity for implantation. So here is a quick recap of all the previous stages which was happening, and now when we arrived at the uterine cavity, we can see that this is the 16-cell stage or morula. Now this morula will be ready for implantation at the uterus wall. So here is this outer cellular mass or trophoblast, while here is this inner cellular mass or embryoblast. When it reaches the uterine cavity, a lot of fluid from the uterine cavity tries to enter into this morula. The fluid tries to enter into the structure, and the central embryoblast is pushed to a side, and another cavity called blastocyst cavity is formed. At this stage, the cellular structure is now called blastocyst. Another important incident that happens at this stage is the disappearance of the zona pellucida layer. The zona pellucida layer disappears, and it gives rise to a number of molecules on the surface of this trophoblast called L-selectin molecules. These cells have receptors which are present on the uterine wall. These receptors are known as L-selectin receptors. An attractive force is developed between these L-selectin molecules and L-selectin receptors. This force is driven towards the uterine wall, and that causes the initial implantation. With this initial implantation, the first week of embryonic development comes to end. Permanent or fixed implantation will happen in the second week of development. This was up till the first week of development. Let's have a quick recap. First, cleavage occurs when zygote is divided into two-cell stage. This two-cell stage is further divided into four-cell stage, then 8, and then 16 called morula. 16-cell stage, compaction process happens, and as a result, trophoblast and embryoblast layers are formed. And with that, fluid enters and blastocyst cavity is formed. This stage is known as blastocyst, and then the blastocyst tries to implant itself to the uterine wall by the help of L-selectin molecules and L-selectin receptors.